Great, thanks. Oh, Sean, do you have another follow-up, or is that a vestigial hand? Oh,
sorry,
down now. That's okay. Yeah, I know. Sorry, Teams is weird. You have to like put your hand down actively. So I guess we're gonna move on. I think Chris, you're taking over.
Chris is going to talk about the modeling work. I'm going to get to our slides for that. All right, go ahead, Chris.
Thanks, Laurel. Hi, guys. Yep, I'm Chris. I'm a fisheries ecologist who's been working on some of the modeling work here. And I'm just going to touch on, first off, a few of the basic sort of considerations that shaped the way we approached this. You guys have already touched a bit on the idea of spatiotemporal modeling and the importance of accounting for space and time. And that's something that we are definitely doing as well. Going into this, we decided to really try and account for the role of ontogeny as well. The fact that species, different life stages of species often have distinct niche requirements. And so whenever data was available for the species we modeled, whenever maturity data was available, we broke them down into juveniles and subadults, or adults and subadults, to try and maybe better resolve some differences in stage-specific differences in habitat use and or niche shifts. We took the approach of trying to use a joint species distribution model, which I'll talk a little more about later, but it's we developed our own approach to this. Most JSDMs, as they're called, use a Bayesian fitting approach that can be really time consuming when you get into large sample sizes and large numbers of species. And so we've kind of adopted our own way of doing this. But one of the benefits of going that route is that you both model species responses to the environment and also their sort of covariances with each other. And so when you predict from these models, you can, especially if you're looking at community level metrics like richness or diversity, your predictions take into account that covariance, residual covariance between species that hopefully provide you with some more realistic predictions. And one other important factor was we really tried to choose covariates that were dynamic, that reflect the the very dynamic nature of this environment, and also predictors that can really be linked ecologically to animal function. And I'll touch on that a little bit more later too. Next slide, please. So again, the modeling approach we used is we called it the CBFM, Community Level Basis Function Model. Again, it models both species responses to the environment and also their covariances with one another. The methods manuscript is coming out this summer. We have an R package that's available now on GitHub. If anyone's interested, reach out to me and I'm happy to help people try and navigate it. It's a pretty cool tool, I think. Next slide, please. So it's kind of somewhere between, if anyone's familiar with joint species distribution models, most of them take the approach of the latent variable approach. So species covariances and sort of spatiotemporal random effects are modeled via latent variables. And rather than, in this case, what we've done is we've kind of tried to replace the latent variables with spatiotemporal basis functions. So in the same way that a species response curve is described as a linear combination of basis functions when you're fitting a GAM, we describe species covariances with one another or their residuals in space and time as a linear combination of spatial or temporal basis functions. So they kind of take the place of the latent variables in a latent variable model. Next slide, please. And just to kind of better illustrate that idea, anyone's having a hard time wrapping their head around it. So if you have, say, a spatial random effect that you represent via a latent variable, that would be this big block on the left. You can see how you might describe that as a function of these other 4 different spatial gradients that are these essentially arbitrary gradients. But when you weight each gradient for at a given observation, you end up with the spatial field that you're trying to represent. Hopefully that makes sense. But essentially what it is a big, giant sort of hierarchical GAN. Next slide, please. So where we are right now, we've fitted for most of the species of interest here, which are demersal, pelagic, and benthic species, about 55 to 60 species with 95 life stages, like species stage combinations. We're training the model right now on 15 years of data, about 10,000 observations from the spring and fall bottom trawl surveys, NIMS bottom trawl surveys, and then testing, extrapolating to the 2016 to 2019 time period. We've got a whole suite of predictor variables that include the usual physicochemical variables like temperature and salinity. We're also, in trying to stay away from more static covariates like depth, replacing depth with some other variables that, again, are kind of more ecologically meaningful. So hydrodynamic stress, how much does water move near the seabed in a given location, and or what is the color and intensity of ambient light in a given location. Those are things that closely covary with depth, but have more ecological meaning, I think, for most animals. We also include benthic habitat descriptors, as you guys, as our Canadian counterparts have described already. I don't know if ours are as good as theirs, but we're working on it. So we fit several models, presence-absence models, as well as negative binomial and a hurdle negative binomial model, because a lot of the species are very over-dispersed, and the hurdle helps to deal with that. And then in addition to the covariates, we have our spatiotemporal component, which includes a sort of, within a year, a time evolving spatiotemporal effect that accounts for differences from spring to fall, and then a separate long-term temporal effect to try and model longer-term fluctuations in species abundances. Next slide, please. So I'm not going to get into detailed descriptions of any predictions, but here's just an example of sort of how the models are predicting. This is summer flounder adults, and you can see, so in the 2 columns, there are spring and fall predictions here. So you can see a pretty clear difference between the spring and the fall. And so the true observations are within each of the spring and the fall panels. The true observations on the left, the predicted are on the right. And then down below are the predictions for juveniles, which are tough to read actually on the screen, sorry. Point being, it does a nice job so far of resolving some of these seasonal differences as well as longer term shifts. Next slide, please. And as Tori outlined, all these model outputs then all go up onto her app, where you can look at everything from the predictions to the covariate effects. The optical niche for the species is that response surface you see in the lower right-hand corner. The estimated vessel effects, how the gear shift, the shift in vessel sampling gear that occurred around 2009 affects each species, and then a long-term time trend for each species gets estimated as well. And next slide, please. So another thing we get out of this is, how much variance is explained by each environmental predictor, as well as the spatial and temporal basis functions. So this is a slew of species. You can see right here, each color represents a different predictor variable, and each of the columns is a different species. And again, obviously, this gets at what are the factors that are most influential in driving each species distribution. You can also look at this across species as well. Next slide, please. And kind of the last thing you get out of this that you wouldn't get out of a traditional stacked SDM approach is there's correlations between species. So you can look at residual correlations or partial correlations. The partial correlations are just kind of an inverted residual correlation matrix that helps to account for indirect interactions. But it's an interesting tool. It helps with your community level predictions. So if two species tend to co-occur more frequently than they might, based purely on the environment, then that gets taken into account in the model when you predict. So that again, when you're looking at community level metrics, like for example, the richness metrics that Laurel highlighted for the State of the Ecosystem report, that residual correlation matrix goes into those predictions and hopefully helps to improve them over what you get out of a stacked SDM. And so you can also hopefully gain some ecological insights out of some of these, and in terms of either identifying if there are missing covariates, identifying dispersal effects, or potentially biotic interactions that are occurring between species. For example, if you have predators tracking prey or competitors excluding one another, things like that, those residual correlations can theoretically can show up in this matrix. So that's a quick outline of the modeling side of things. I'm sure people have questions, but I'm going to leave it at that for now.
Thanks, Chris. Does anybody have any questions for Chris? Raphael?
Hi, can you all hear me clearly?
Yes.
Yeah.
Great. My name is Raphael. I'm a PhD candidate in statistics at Dalhousie University at Dal, and I was curious. You put a lot of like oceanographic predictors, and I'm wondering, are these like direct observations, or are they coming from like oceanographic models and that type of stuff?
A lot of them are coming from reanalyses like GLORIES, and there's a more local reanalysis that I'm looking at using called Doppio that's fits to our region. So yes, a lot of those oceanographic variables are coming from reanalyses.
Cool. And I was wondering if they are, how worried are you? And well, if like, have you looked into like potential error propagation from those models forward? I'm aware this is generally very difficult to do, but I'm more asking out of curiosity.
Yeah. So there's been a bit of work looking at GLORIES in particular and how well it represents things here. And most of the work that I mean, there are fine scale biases for sure, but it's pretty representative. But we were also, for some of these fitting models, comparison models, using the observed observations at those at the actual tow locations, because there are salinity and depth temperature profiles recorded at each tow as well. So we have that data to reference, go back and forth between.
Well, thank you.
Great, thanks. Any other questions? Here's something that I will. Okay, great. Thank you so much, Chris and Laurel and Tori. We'll see you back at the discussion and wrap up. But I guess we're moving on to the Canadian